Welcome to today's webinar. My name is Steve Oldenburg, and I'm the founder and CEO of Nanocomposites. Today I'm going to be talking about nanosafety, conclusions from a decade of nanotox research. For over 10 years, we've been helping supply material to toxicologists so that they can understand the effects of nanoparticles on the environment and human health. Our role is not only to supply the materials, but also to help toxicologists integrate those materials into their experiments. This is important because with nanoparticles, it's not just about ppm exposure levels. There are many other factors that need to be incorporated into an experimental design in order for the data obtained from the experiments to be valuable and reproducible. Since the inception of nanotechnology more than 3 decades ago, there's been a concern that nano was somehow inherently dangerous. This was propagated by books such as Prey by Michael Crichton with its self-replicating nanobots, but also throughout the popular media where scientific results would get picked up by the news outlets, often reporting results that didn't have the right context. All of this interest led to the inception of a new field called nanotoxicology that was dedicated to determining the risks associated with nanoparticles and their impact on the environment and human health. Now, it turns out that exposure to small particles isn't new. We've been exposed to nanoparticles since the dawn of time. The vast majority of nanoparticles released into the environment are due to natural or industrial processes. Originally, this research was called the study of ultrafine nanoparticles. About 20 years ago, it got reclassed as the study of nanoparticles and became the field of nanotoxicology. Now, what's new is that as things advance technology-wise, the production of larger quantity of engineered nanoparticles have the potential to be released into the environment. And this has led to a new focus on understanding the safety of these emerging materials. So to determine safety, we have to consider a number of factors. Ultimately, the goal is to build a set of structure-activity relationships that can predict the risk associated with an untested nanomaterial. We start with the physical-chemical properties of the material itself. This is the physical-chemical ID. And there's a whole bunch of components that are important to understand that are different than the traditional chemical characteristics that toxicologists usually incorporate into their experiments. Once we have the materials, we can design experiments to look at their hazard. And this has a number of integration challenges because nanoparticles behave differently than traditional chemicals. Finally, we've got exposure. Ultimately, if we're not exposed, we're not at risk. And so we have to understand what are the unique characteristics of nanoparticles that might impact our exposure. And ultimately, how do we get from exposure to hazard and risk and really understand what what's going to happen when these particles are released in the environment and what do we need to be concerned about.
Like I said, the history of nanocomposites goes way back to 2007. There was an early Air Force SBIR that was sponsored by Dr. Saber Hussein at the Air Force. And what he recognized was that in this emerging field of nanotox, there was a problem. There were lots of groups reporting results that were very inconsistent, and it was making it difficult to determine where to focus effort and how to move the field forward. And one of the main problems that he identified was the lack of consistent starting materials. So the materials, people were ordering them from Sigma Aldrich and getting dried powders and sprinkling these aggregated dried particles on cells, and then reporting some results. And of course, those results were very different and somewhat confusing. So the idea behind this SBIR was, can we create these libraries of very well-controlled materials that are going to allow the researchers to get relevant data that's going to be reproducible, and we can start to make some connections between the physical properties and their ultimate toxicity and risk. So we had this library that got built up with precisely controlled different sizes, different shapes, making sure things were unagglomerated. Spent a lot of time purifying and concentrating materials to make sure that the effects were just from the nanoparticles themselves and not from residuals, making more multifunctional or complex materials to tease out some of the nuances, and then overlaying that with this extensive characterization. So the particles that we focus on, which is really just a subset of the nanomaterials that are of interest in the nanotox space, but it's mostly in the metals and metal oxides. And here we've got a core materials of gold and silver. Some of the plasmonic materials have very precise and interesting ways of controlling their shape and optical properties. Materials like iron oxide that are magnetic, titanium dioxide, which is high index and photocatalytic, and then we've got some other materials like silica and and copper oxide that are used as, in some cases, antimicrobial, and in some cases as sort of a structure or scaffold to put other elements on. We can coat those with a particular surface, and we can overlay that with an actual shell. So we can build up these core shell layers that have different properties as well. And then in addition to spherical properties or spherical particles, we can also do all of these different shapes. And each one of these components actually is quite important. And when you change something in this construction, you can end up with a completely different biological interaction. So sort of libraries of where experimenters are able to get these vectors of just changing one thing at a time are pretty important for kind of understanding what's going on and moving forward. So the benefit of the SBIR was that we were able to really provide materials and then interact with the toxicologists. And so some of the early experiments, this was some work done by Nancy Monteiro-Riviere, where we had provided them some materials, they had seen some toxicity, we had tried to tease out what the reason for that was, and it turned out that there was some residual reactants in that initial solution, and once we purified it, that toxicity went away. So that was, very early on and really emphasized the importance of purifying. We want to understand that the effects that they're seeing are from the nanoparticles themselves and not from something else from the manufacturing process. So that led to us, not only doing purification, but measuring for endotoxin, trying to keep things sterile, all of the other elements that could be convoluting or affecting the assay besides the particles. The other issue, which is what we refer to is transformation. And the problem here is that the nanoparticles aren't static. And this is a little bit unusual from what people were used to using it in toxicology, where you've got this chemical and you had to add it at the certain parts per million, and that was really what you had to worry about. Here, the nanoparticles have a whole bunch of different characteristics. So we're trying to make sure that that's all going well, and that we sort of understand how these things are being altered with time, because we need to make sure that everybody's starting at the same spot. So one of the ways that we got around that was for some materials we used silver nanoparticles that were in a dried format. And previously we had told people that drying components was not a good idea because the particles would be permanently aggregated. But we found a way to lyophilize them, and then they can just be, you can add water and they'll sort of pop right back up and be in an aggregate state. And this was used by NIST, which had this problem because they certainly were being required to provide a spec sheet that was absolutely reliable, but if they used liquid nanoparticles, they would change with time. So what they would do is we'd start with this freeze-dried formulation, provide sort of a ground zero after dissolution, and then things could be characterized after that. So in terms of going forward, then that gives us this sort of the physical chemical characteristics that we needed to know. But the question that the toxicologists were asking was, well, what are the things that are different? Why, what do I have to be worried about, or should I be thinking about when I'm designing my nanotoxicology experiment? And there's a number of factors that make nanomaterials different than the standard chemicals. And I'm just going to run through them quickly to give a kind of a flavor of the extra things that people need to be thinking about. So in terms of the most obvious one, which is just size. So there's a number density and a surface area per unit mass that increases as a function of size as these particles get smaller. So when you're down in that 100 or 10 or even down to the 2 nanometers, the number of particles per unit mass is greatly increased. The surface area is greatly increased, and that's something that we have to think about. And then the size also has a dependency for fate and transport. These particles are on the order of the size of a virus. They're going to be handled by biology differently than some particles that are much larger. The second thing is surface charge. So the particles themselves, when they're suspended in solution, have a a double layer that keeps them stable, repels them from each other, and allows them to remain individual. And that surface charge will, if it's a negatively charged particle, it's going to be attracted to positively charged surfaces or positively charged particles that they could potentially interact. So that surface charge is one of the primary ways that we think about how these particles interact in a solution. We've got the surface coating, which also impacts the surface charge, but has other functions. It can sterically keep the particles stable, and in some cases the surface coating can be designed to target or recognize certain things in solution if it's coated with, for instance, an antibody or some other biological targeting moiety. The other things that we can look at are composition. I had mentioned earlier that silver was relatively dynamic. It has a dissolution constant in water, and over time it can release ions. And in the case of silver or copper, those ions are toxic. So it becomes it's not necessarily that the nanoparticles themselves have a very high toxicity, but it could be that the ions that they're releasing in that local environment do impact the toxicity or the effect of them on the environmental biological system that's around them. The morphology is also important. There's asbestos was a good example of a structure that became much more hazardous because of its shape. And there was a concern in the nanotoxicology space that the same thing would happen with nanomaterials. And that was true to some extent, but in most cases it wasn't as dramatic as the asbestos example. So here we've got a wide range of sizes and shapes that can all affect surface absorption, stability, transport, and uptake. The band gap, this is a sort of a select group of materials, but is important. So sometimes with materials with a band gap, if they're excited, they can that can impact their optical properties, the redox properties, or the reactive oxygen generation potential of the material. Very small particles are used in catalysis, and that's because they have surface area and nanoscale structure that they enhance the reactivity of certain processes. So that enhanced reactivity, either at just because of that surface area or at edges and corners, can drastically affect how the particles interact with different compounds. Plasmonics is one of the things that we do here. This is where we're using materials, mostly gold and silver, that have very strong interactions with incident light, and that can magnify the local electromagnetic field, again, influencing some of the characteristics of the particles in biology. And then finally, we've got phase stability, where at the smallest sizes, there's a difference between the bulk and the nanoscale dynamics as we change phase. This is most often seen in sintering, so where you've got very small particles, and if you apply a heat that's much below the bulk melting point, you can still get the particles to stick together. And this just has to do with the high curvature and the high surface area of the materials themselves. So and then one more is defects. So we've got synthetic materials that are that can be influenced by having an impurity in them, and that impurity can actually lead to the last one, which is instability. So here you can have these small particles that can change with time, and again, this is another concept that isn't usually seen in the bulk field, where due to that curvature of the particles, they can release ions, and that the particle size distribution can change with time. So it's almost like you had a bucket of marbles with very large marbles and small marbles, and the next day you'd expect to come back and that size distribution would be the same. And with nanomaterials, sometimes that's not the case, and sometimes you can have the smallest particles going away at the expense of the larger particles. So all things that, if you were coming from a straight toxicology background, you wouldn't necessarily be thinking about this sort of level of change or dynamics or range of properties within the chemical that you're studying. So this gets us back to our sort of overview, and we've got this idea of the physical chemical properties, and we've got all of these these nano-specific things that we're going to worry about or be concerned about when we design our experiments going forward. And so that's really going to provide a framework for sort of designing what happens next and where we need to go. So in terms of the experiments themselves, we've got there was quite a bit that was new for integrating. As we went from sort of fundamental research to the ultimate goal, which is predicting a biological response from these nanomaterials. And we've got all these data gaps and some challenges associated with filling those data gaps. So again, and some of those problems, I won't read them all off here, but we've got some instrumentation, some analytics challenges, measuring the particles when they're in these complex biological matrices. We've got dosing issues. We've got new protocols that, for how to handle these materials and their aggregation state as a function of time. And so all of these problems are sort of things that the community was trying to address over the last 10 years, and literally 1,000+ papers kind of looking at the impact and influence of nanomaterials in these biological systems. And in this talk, I'm actually not going to talk so much about the results of that, but I did want to highlight that we are making progress, and that this is a paper from Dr. Saber Hussein's group, and so here they're finally getting to make some predictions or some generalizations, design rules really, for how these different characteristics of nanoparticles affect their ultimate toxicity. So if you read the paper, they talk about size being definitely a trend. So decreasing the particle size generally increases the toxicity and the amount of cellular uptake. For charge, there's a generalization that in most cases, positively charged nanomaterials are more toxic due to their increased interactions with primarily negatively charged biological surfaces and entities. From a composition perspective, if there was a rate of ionic dissolution and those ions were toxic, there was definitely a correlation between that and the ultimate toxic result. And in morphology, where rod or anisotropic shapes were taken up less efficiently, but once they were there, they could introduce more significant damage. The reason why this is so important is twofold. One is that we might be able to make some estimates or some predictions of a new material that we haven't studied yet on what its toxicity might be if we have these design rules. And the other one is that we can also use these rules to design safer materials. For instance, in nanomedicine, it's really important that we understand that the things that we're injecting into the body are going to go to the right place and aren't going to cause damage. So there's a whole sort of secondary field coming up with nanosafety and really thinking about how to make sustainable nanomaterials for safer design of components that could ultimately end up in nanotherapies and be used for treatment. So one of the themes that I try to present when I talk to the toxicology crowd is that while they're out sort of looking for things to protect the community or to identify risks, there's also this other side where they're providing a foundation for some of these future nanomedicine, nanobiology type interactions where the data that they're obtaining is actually very interesting for that community as well. So going back to our sort of map of how to delegate risk, the last component I haven't talked about is exposure. So most of the experiments to date have been done with pristine systems, and because you really want to try and find out what's going on. And if you start with a very complex material, it's very difficult to determine which component of that matrix or that complexity is causing the effect. So there's been a lot of pristine studies, but that's really not what we're exposed to. We're exposed to materials that are integrated into composites. We're exposed to nanomaterials that are potentially made with uncontrolled, unpurified processes and are very complex. So it's important to recognize that some of the results that are coming out of the pristine experiments might not be completely relevant to an actual risk because it's just unlikely that that's going to be an exposure route. So if we take that a little further, we can look at where the split has been in terms of the studies that have gone on, and it really has been dominated by that hazard spot. So we've got a few Most of the studies in hazard, we've got some fate and transport, and then there's a smaller component that's really due to this exposure question. And you can think about exposure. You have to really think about the life cycle. So, like, what does exposure mean? Well, it can mean different things at different stages in that life cycle. So from a manufacturing perspective, we've got, the handling and the making of the particles themselves. There might be some injection molding process or some nanocomposite process of processing things to get things in. And then once it moves into commercial or consumer use, then there's a whole other range of elements that could happen to that material. UV degradation, thermal degradation, hot or cold. We could get some leaching, some biodecomposition. There's some mechanical elements to that. We could have abrasion or deformation or sanding or drilling. All of those things might release a particle. And then we've got more end-of-life type of consideration. So once that item's been discarded, is it going to be recycled? Is it going to go into some sort of a landfill? If it's in a landfill, is there a degradation process there? If it's incinerated, is there a possibility that there could be something that would be released from that thing? So when we're talking about exposure, we've got to kind of really think about this much more complex degradation process and life cycle. So if we take a concrete example of, so for instance, a carbon nanotube reinforced composite polymer, and the carbon nanotubes are being added to increase the strength, but there's a concern that what happens if those particles get out? What happens if carbon nanomaterials are released? And that could happen presumably by weathering, and you might lose some of the resin, and you would get these carbon nanotubes that could be individualized and potentially be an exposure source. They could be mechanically ground down where you got them small enough and you would release the carbon nanotubes themselves. So sort of the question is in a composite like environment like that, what is the exposure risk with given a particular life cycle of a material? So this was Stacey Harper's work, and she looked at a whole slew of different composites and nanomaterial or nanotube materials, and they examined them, and what they found was that while you can see the nanoparticles or the nanotubes in these particles that are released, they basically, across more than a dozen different studies, didn't see any free carbon nanotubes being released. So, and this has been sort of repeated in a whole bunch of different things. So it's just really not that common when something is integrated for those particles to be released individually. And that's true for composites, and it's also true for just solutions of nanoparticles. We spend an enormous amount of time at Nanocomposites trying to keep our particles from aggregating, from coalescing and getting bigger or depositing out or plating, because that's almost their natural state. That's really where they're going to go unless you're careful. And so that tends to be what happens in the real world. Things will start out in a nanoform, but over time they're generally going to end up in some sort of a larger composite that's going to be less likely to have some of these specific nanomaterial dissolution and transport issues. So where does that leave us? So we've got a lot that's been learned on the particle side, much better particles that are being characterized. That knowledge of what's important has been transferred to the toxicologists. They've done an enormous amount of work on understanding the effect of the individual nanomaterials, and then we're just now taking that data, incorporating it into real-world examples for exposure ID, and coming down to a potential risk. And the conclusion, which is good, is that, this is a quote from some work from Dr. Gracian that did sort of a recent review paper in Environmental Science Nano, and basically says, to date, no known human diseases or serious environmental impacts have been reported that are specific to engineered nanomaterials. So no doubt that's good news. And I guess as a kind of a follow-up of that, what was interesting is I was at a GRC conference on environmental nanotoxicology last year, and there was a session with what I thought was kind of a surprising title, and that was the challenges of reporting null results. And when you got into the session, what the what the topic was that a lot of the materials that have been studied really didn't have a huge effect on toxicity. There's just a lot of null results. There was a lot of things that didn't have anything surprising associated with the nano element in terms of what you would have expected versus a bulk material. And the reason why I thought this was important is that they were having trouble getting those results published. Basically, the reviewers were saying, well, yeah, that's great, that's not interesting, and sort of move on. And my comment to that community was that some of those null results may be the most important element of the work, in that a null result in nanotox is really a positive result in nanosafety. And by understanding what surfaces, what sizes, what shapes don't influence or don't elicit a toxicological response, provides us with a working set or some guidelines on how to build these next generation delivery mechanisms and other elements of nanomedicines and nanotherapeutics that are going to be extremely important in the future. So, are we out of the woods? And I think the obvious follow-up to that is absolutely not. But we have learned a lot, and there's a couple of sort of big points that I kind of just wanted to emphasize and make. And one was that the first thing is it's difficult to find examples of common processes that will release large quantities of engineered nanomaterials. Partly that's because there's just not a huge manufacturing industry that's associated with producing those materials right now, but it's also that a lot of the materials that would be risky are getting integrated into something else that just doesn't have that potential for release. The second thing is that if the nanoparticles are released into the environment, they're typically rapidly aggregated, encapsulated, or otherwise sequestered into larger, safer materials. And so that's just generally good news. And then the third thing is that the risks that are identified by many of the in vitro experiments, if you look at it and you extrapolate back from the exposure things, it's a little bit unrealistic. So even the results that are coming out of literature that yes, yes, this is toxic at this particular dose, and that level of toxicity is increased over the bulk, the actual risk or hazard associated with that from an exposure perspective is relatively low. So where does that leave us? Well, I think we're really just getting started on this. The overall conclusion of the research from the nanotoxicology space is that it is incredibly complex, to the point where we're never really going to fully understand it. But that said, we are learning a lot, and the things that we're learning, I think, transcend just the toxicology space in terms of our understanding of basic fundamental processes of materials interacting with biology. Very subtle differences in the properties can result in really surprising changes in vivo. Very slight changes in the size or the surface state can completely change the protein corona that gets absorbed onto a particle once it's introduced in vivo. There's huge challenges remaining in post-transformational characterization. How do we understand how these particles are changing once they're in the system? We have pretty good tools for understanding the particles before, but it's once they're introduced into a complex matrix, very difficult to figure out what's going on. There's all kinds of surface dynamics that are going on that are complicated and hard to really drill down to. There's transport issues, there's elimination issues, and then there's the global problem of extending these results from the pristine materials to much more complex industrial byproducts. Continued nanotoxicology research is going to be absolutely essential for us developing safer nanomaterials now and in the future, and then also providing this backbone for the nanotherapies and the nanomedicine that's coming down the pipe. So with that, I'll wrap it up, and then I'd be willing to take any questions that people have about the talk or nanotoxicology in general.
Great, thank you, Steve. I'd like to thank our audience too for your attention. We're going to take some time, as Steve mentioned, just to kind of address some of the questions that were submitted. If you still haven't submitted a question and you'd like to, please go ahead and type your question into the question pane, and we'll try to go ahead and address that today. Our first question comes from Sahir. What are the regulatory challenges in incorporating nanomaterial into commercial products?
Okay, yeah, great question. So the FDA's take on nanomaterials is that there really isn't anything fundamentally different about how they're going to treat nanomaterials from how they've treated every other compound that's going through the FDA. And with the sense that what you need to do is you need to establish a reproducible process for making those materials. And once you have that reproducible process for making the materials, then there's a well-defined system for going through the safety and then the tox and then eventually the distribution of how it gets distributed in the body, and then we can go forward and make an assessment on whether the material is safe or not. So there's so while the FDA is still trying to understand maybe some of the nuances of those analytics and those methods, there's really nothing fundamentally different than make it reproducible, study it, and see if it has any adverse reactions before you scale up and go to a real full efficacy clinical trial.
Thank you, Steve. Our next question today comes from Nancy. Does interactions with cells and protein affect how nanoparticles behave in biological systems?
Yeah, so definitely. So the particles will start out in one state, and as soon as you introduce them into a biological system, they are different. And the first thing that happens is typically there'll be proteins or other elements that are in solution that'll associate with that surface. And so now you've got a particle with a completely different surface. It's got a different charge, a different hydrophobicity, and that's going to control basically its transport and its ultimate dissolution wherever it ends up. So the complexity of it is that we start with one state, it gets transformed, and it's in sort of this constant flux as it moves into different compartments or gets exposed to different environments while it's in the environmental or biological system of interest.
Thank you. Our next question today comes from Ben. Is it possible to purchase a bare surface of your particles? And then would this be less toxic?
Yeah, so that's a great question. We get that all the time about naked and nude and bare nanoparticles. So a particle it always will have a surface, and what people typically mean when they ask for bare particles is they want something that's stabilized with a molecule that's as easily displaceable as possible. So what happens is that you've got a metal surface that's got an affinity for things. There's other stuff in solution. Things are going to associate, and if you don't have anything stabilizing the particle, then they will typically just aggregate and fall out of solution. So when we say bare, we think about what is the lowest affinity, smallest perturbation molecule that we can have on the surface that's still going to have it perform or remain stable as a nanoparticle. So our carbonate materials or our citrate materials are really the closest thing that you've got to having that sort of naked state or bare state. And typically we'll start with that, and we'll use that nakedness or that that low affinity to build up different surfaces. So if we add something with a higher affinity, that'll displace the citrate or the carbonate, and that will impart a new surface to the particle that has some other functional characteristic that's important and interesting.
Great, thanks, Steve. Our next question today is, are silver ions more toxic than silver nanoparticles, and can you distinguish, are they indistinguishable from each other?
Right. So there's So in terms of mass loading, silver, so like silver salts are more toxic than silver nanoparticles, and that's shown over and over again. And that, yeah, the silver ions themselves have, in some cases, an order of magnitude higher toxicity than the silver nanoparticles from a mass basis. That said, the silver nanoparticles are a high surface area source for silver ions, and so by having silver nanoparticles, what can happen is you can transport those nanoparticles to a location, and then they will basically be continuously releasing the silver ions. And the second part of that question is, well, how do you tell the difference? And we have a number of ways of doing that. In some cases, we can measure in a given solution of silver nanoparticles. There's going to be a certain fraction that are in the solid silver nanoparticle state, and there'll be a certain fraction that are in the ionic form. And so it's possible to measure that. And typically, what we'll do is we'll either hard spin or filter out the silver nanoparticles, and then we'll measure that supernate or permeate from the filters and do ICP mass spec, and then we can get this split between what the silver ion concentration is and what the silver nanoparticle concentration is. And one control that some nanotoxicologists do is they figure out what that split is, and then they'll actually add, as a separate experiment, the same amount of silver ions into that solution, and so then they can tease out what's the ultimate effect of both silver particles and silver ions, and then just the silver ions at that concentration alone to learn something about that system. There's a new technique of single particle ICP mass spec, and that's where you can launch individual particles into an ICP mass spec, and it'll give you actually that split, that solid particle concentration and that ions solution in the same measurement.
Great. Our next question comes from Anthony. Why is it that positive nanoparticles are more toxic than negative ones?
Yeah, I think it's complex. Like quat ammonium, compounds are typically used for antimicrobial type of things, and I think it has to do with sort of the effect of how some of those cations are interacting with materials. The basic thing that's seen is that it just, most things in a negative or in a biological system, for whatever reason, have a negative charge. And so if you've got a positively charged particle and it can actually stick or interact with the element of interest, then typically that particle, now that it's on the surface, has a higher chance of uptake, or if it's releasing silver ions, now it's releasing silver ions very locally to the surface of that entity and can induce a higher level of toxicity.
Great. Well, I think that concludes our question and answer section for today. I'd like to thank you, Steve, for sharing this valuable information to our audience, and I'd like to thank everyone again for attending. If you have any additional questions that you did not ask on our live recording, you may reach our technical support at the email provided on the screen, which is info@nanocomposites.com. And on behalf of Nanocomposites and our presenter, Dr. Steven Olderberg, I'd like to thank you so much for joining us today.